All right. Hi, this is Megan Baldridge. I will be giving 2 lectures on metagenomics. So this is obviously the first. These were originally scheduled for March 23rd and 25th. Obviously, you can be in touch with the TAs about appropriate times to listen to these lectures as needed. So an important disclaimer, in these highly chaotic times, mostly just going for good enough in this lecture, so I apologize if there are any errors or awkward pauses. I would also, of course, like to give an enormous thank you to Gautam, who has previously given these metagenomics lectures and allowed me to borrow very heavily from his previous lectures. All right, so in general, of course, we're going to be talking about metagenomics here, or the genomic study of uncultured organisms. So in giving this lecture, I'd just like to start off by saying I know many of us have had our lives really massively disrupted by one particular microbe. So you may be feeling a little bit unenthusiastic about microbiology, very understandably. But hopefully you don't need to be convinced that it is still, of course, fundamentally important. And also that better defining microbial genomes, either as single genomes or in complex communities, is a rapidly evolving area of very important research. So in general, metagenomics includes both the research field and also a set of research techniques that are used to actually probe these complex communities. So in focusing kind of on the research techniques, in general, these are experimental and computational methods meant to overcome the really massive complexity of microbial populations. So as we'll be getting into, these different microbial populations are extraordinarily diverse. Many of them are extremely difficult to work with. And so we have really come to rely heavily on the capacity to interrogate the genetic information that we can gain from these communities in order to ideally maximize our understanding of both the compositions and the activities of these communities. In general, we're trying to kind of representatively sample the complexity that cannot really be exhaustively characterized, but we're at least trying to get as much information as we can using the techniques that we'll get into. As a research field, I think it's really important to remember that for metagenomics in general, this is focusing on understanding biology at the aggregate level. So in the context of a complex community, multiple organisms might be contributing to any particular function, an effect, or a phenotype. This could include something like generating some particular factor that interacts with the immune system, or it could be utilizing a set of nutrients. So any kind of output or input may be actually mediated by multiple organisms acting together. So of course this means that these functions can then transcend individual organisms, and a lot of metagenomics research is focused around describing community function. So in general, it's important to think about understanding not just necessarily single genomes, which are important, but also networks of genes that are working across different organisms to serve these collective functions. So in general, it also is kind of a systems biology for communities and also the biosphere. All right, so I'm just kind of thinking about this from a more historical perspective. Really classic thoughts of, what is the importance of microbes, really go back to Koch's postulates, and just to kind of review these. In general, the idea is that if you're looking for a microbe that's mediating an effect, specifically causing disease, the way to actually show that that microbe is responsible for causing the disease is that you must find it in abundance and all the organisms suffering from the disease, but not in healthy animals, but not in healthy individuals. This microorganism must be isolated from a diseased organism and actually be able to be grown in pure culture. This then cultured microorganism should actually be able to cause disease when introduced into a new, previously uninfected organism. And then you should hypothetically be able to re-isolate the microbe from this second experimental host and identify it as being the same to the original specific causative agent. So, this set of postulates has been around for quite a while and has really been kind of the classic goal in microbiology for identifying pathogens. And, the challenge with this way of thinking about how microbes are actually interacting with hosts then led to what for a long time was really kind of this culture-based paradigm. So exenic cultures, which are free from any organisms other than the one that's actually the goal species, so a pure culture, have traditionally been thought of as the gold standard for good microbiological practice. If there are multiple species in a culture, that's long thought of as more of a contamination as opposed to potentially a community. If you think about things like biofilms, then these are classically thought of as sort of accidents and not really of any use for scientific study. And traditionally, genomics kind of really reinforced the use and the need for pure culture by being able to actually permit more sensitive analysis of the presence of contaminants. And so for a long time, there was really this idea that if you wanted to study a microbe, you needed to be able to cultivate it, and then you really could then be able to do any additional future studies. So in getting at this, classically, when we think about how microbes are studied, it's usually by culture-dependent methods. So microbes are classically plated. You can see petri dishes, which contain agar as a solid growth medium, plus key nutrients, or you grow microbes in different liquid medias, aerobic or anaerobic conditions. But a really key important thing to remember is that the majority of bacteria in most habitats are really not easily cultured. So, there's kind of this, the most classic examples is that Van Leeuwenhoek, who's really thought of as the father of microbiology, just, took out some plaque from his own mouth and looked at it under a microscope, and there saw this enormous diversity of different bacteria that even just to the naked eye using a microscope he was able to see these really different morphologies. And this is really the case for kind of almost any surface or habitat that you would be looking at. But the challenge is that, almost all of these cannot be readily cultured. So different microbial communities are present, again, any surface or area that you can think of. There are some microbes present there. Really, of course, there are some sterile sites, but even in places that are thought to be fairly clean, you might think of, hospital surfaces or the air as places that maybe shouldn't routinely be full of microbes. But indeed, all of these different habitats have their own different microbial communities. And of course, these different microbial communities are all highly interconnected. And so, they are, of course, mediating then effects on different habitats. Here you can just see one sort of example wherein you can think of this as how antibiotic resistant bacteria could be actually transferred around habitats. So if food animals are fed antibiotics and develop antibiotic resistance, which is certainly the case. You can end up with those bacteria ending up in the soil, runoff ending up in rivers, seas, and lakes, and then being passed along to humans via drinking water or swimming, and then this kind of other complex transit of antibiotic resistance through different habitats. And it's just kind of one example, but of course it's an incredibly complex interplay. And so, for the purposes of both this talk and then kind of actually what's really been studied generally for metagenomics, bacteria have been really by far the best studied component. But I would just like to point out that, of course, in addition to the really complex communities of bacteria that are present in any given habitat, there are also fungi, sometimes helminths, as well as really complex communities of viruses, both prokaryotic and eukaryotic, that are, of course, also present at those sites to varying frequencies. And so this is just showing some numbers for human microbiota and macrobiota. But of course, that may vary depending on what particular samples are being explored. But just something important to remember as we're going through is that depending on the methods used, analytical techniques may or may not actually be powered to look at other types of microbes beyond bacteria. And I'll kind of point that out as we go along. Okay, so as we mentioned, if we really want to start to look at complex communities, we have kind of a few different options. The first is to rely on culture-dependent methods. So in that case, what we might do would be to actually isolate members individually via culturing, perform functional testing, such as looking at antibiotic susceptibility, for example, performing sequencing of these single genomes from isolates, or just screening isolates for particular genes of interest by PCR-based methods. Or we could also take culture-independent methods, in which case we would just isolate nucleic acid from a given community and then just directly sequence it. So kind of the reason that direct sequencing has a number of benefits is that fundamentally, so we've already mentioned, there are the majority of microbes are really difficult to culture. And kind of a classic example of that is just shown from, as far back as 1985, where if you just do direct comparison of, in any given sample, if you look at the number of cells that you can see on the microscope, then you look at the number of individual colonies that you can actually culture, there's just a really shocking difference on the orders of magnitude between the number of cells that you actually can see just by microscopic examination, really highlighting just how unculturable most microbes are. So in general, if you're able to kind of bypass the requirement for culturing, it's really ideal. Now, just something to kind of think about when we're thinking about looking at microbes is kind of going back to really sort of basic molecular phylogeny. So in general, kind of a very classic look at phylogenetic analysis based upon ribosomal RNA sequences, specifically small subunit ribosomal RNA. Kind of reveals that living systems really derive from three distinct lines. So there's eubacteria, or kind of all typical bacteria, archaeobacteria, which include methanobacteria, and then eukaryotes. And we'll kind of get into this more, but this really represents kind of the classic tree of life. But importantly, from these three different lineages, you can see that the multicellular lineages, as shown in red, are quite rare and extremely not diverse when you look at this, when you look at rRNA sequences. And really most molecular diversity in the world is actually found in microbes. So in general, I'd just like to highlight, it's really a prokaryotic world dominated by single cell organisms that are incredibly diverse. Oh, and I'll just go back and make one quick point, which is just that these multicellular lineages shown here are actually Zeoges corn, Homo, which is us, and then Coprinus, which is a mushroom fungus. And so, yeah, those are very different as far as kind of we're concerned. But when you're actually looking at that level of diversity compared to then the level of diversity among bacteria or archaea, it's just really shocking how incredibly diverse these microbes are from a genomic standpoint. Okay, so getting back to kind of guesstimates for number of species, say it's still not really known. We can guess in the millions, but certainly there are many habitats and areas that have not yet been sampled, or and even our ability to fully understand the complexity of the different species in our own intestines, even though it's been one of the best studied habitats, we're still uncovering very new and distinct species from even that site. So yes, still much to learn. So in general, there can be just a really shocking number of different species that are present. They can be found in really diverse habitats. So from extraordinarily hot sites, radioactive, deep underground, all sorts of environments, which obviously would not support, quite as many eukaryotic organisms as well. These different prokaryotes have widely diverse cellular chemistries, and they serve tremendously important functions in their environments, in actually processing nutrients, converting different compounds, obviously in human health and disease, and so are just terrifically important. So one thing that I think that's very exciting in general about this field is that our ability to actually look at and start to understand some of this diversity has really been massively improving. So prokaryotic genomes are really rapidly being sequenced. We'll get into a little bit more about why that's been happening so much more over the past few years. But in general, there are now many more prokaryotic genomes that have either been completed or are in progress than there were even 5 years ago, making kind of the ability to both analyze new samples and compare to previously published data, of course, much easier. So kind of another really important factor to keep in mind when thinking about actually how do we analyze bacterial genomes are some kind of distinct challenges present in bacteria that are less of an issue in eukaryotes. So an important thing is that in addition to the massive complexity and diversity present in microbial genomes, these genomes are also constantly evolving. So in addition to kind of being rapidly responsive to environmental pressures and development of, for example, resistance mechanisms by vertical transmission, genes can also be shuffled horizontally between different bacteria via either transformation, and you can just think about how we leverage this capacity of E. coli all the time to grow plasmids. But also phage can actually serve to drive horizontal transmission by transferring genes from one bacteria to another. And then there's also horizontal transfer of genetic material between cells by direct cell-to-cell contact or by these kind of bridge-like connections between two different cells. And horizontal gene transfer in bacteria is extremely widespread. And here, even not just between bacteria, but between bacteria and archaea, which you can see is there's a particular operon, and highly unrelated bacteria actually have these very closely conserved operons. So in this case, you can see that this methanobacterium has an operon that looks extremely similar to one in Clostridium. And if this was kind of the marker gene upon which you were basing your phylogenetic inferences, you would assume that this methanobacterium was perhaps even more phylogenetically closely related to the first Clostridia than the two Clostridia were to each other, when in fact they're extraordinarily distantly related. So this horizontal gene transfer can really make it potentially quite tricky to sort out where in the phylogenetic tree did any particular microbe come from. And, I think this is just an extremely kind of widespread challenge. So even if you're just looking at bacteria that appear to be the same, at least at the species level, by just doing 6-mass analysis, they can be highly divergent both genetically and phenotypically. So here's a comparison of 3 different E. coli's, whereas one is a harmless lab strain, another is a highly pathogenic strain that causes UTIs, or urinary tract infections, and another one which actually infects the gut and is responsible for quite a high fatality rate. And so even though these, just doing 16S would suggest these are the same species, then the rest of the genome is actually quite distinct between these different E. coli's. So, we'll talk about 16S analysis and also metagenomic analysis. But I think that, even for things that seem to be closely phylogenetically related, there's still the capacity to have highly differential activity in different contexts for individual microbes. Okay, so hopefully that kind of provides adequate background for where we're going to dive in now to sequence-based metagenomics. Okay, so I imagine this has come up previously in this class, but of course, applies to the field of metagenomics as well. DNA sequencing keeps getting cheaper and cheaper. The sequencing costs continue to drop much faster than Moore's law. Something important to just remember here, of course, that even if you are sequencing a huge number of microbes, actually being able to assemble and then complete or finish a genome is more costly than just the raw cost of sequencing, because often you'll have to go back and do additional bridging sequencing or figure out ways to do hybrid assemblies using long and short read sequencing. But regardless, it's still infinitely more practical today than it was, again, even 10 years ago. So for metagenomics, there is, of course, a requirement for massive computing resources. So depending on the different technique that you're using, you may end up doing more or less very deep sequencing in order to get good coverage of what might be a number of different microbes present in your sample. So we'll talk about 16S, which is a quite cheap approach to get a sense of the community composition of a sample. But other approaches, metagenomics or metatranscriptomics, can be require really a lot of very deep sequencing, and then of course analysis approaches to actually take the raw sequencing data and make sense of them can be quite intense. Okay, so we're going to focus today on kind of two of the more straightforward ways to look at bacterial communities. So, for any given sample, let's say my lab generally works with mouse poops, of course, environmental samples or other human poops or different sample types can, of course, go into this. So in general, once the DNA has been extracted from a sample, then the options are to move towards a 16S gene-based survey. And so we'll get some more details of that. But in general, this involves PCR amplifying using sample-specific barcodes, sequencing 16S rRNA gene, or a small portion of it, to get a sense, at least, of the community composition. Other options include doing shotgun sequencing of the metagenome or the metatranscriptome. So obviously DNA can be used for the metagenome. cDNA can be made from RNA from the sample in order to capture the metatranscriptome. And those sequences then can kind of additionally help answer who's there, but can also provide a lot of really critical information about the functional effects of those particular communities by giving much more detailed information in the genetic content. Okay, so we'll start off first by focusing on 16S, our RNA-based characterization of bacterial communities. And I'll mention that beyond this, there are kind of very similar approaches using 18S marker gene sequencing, which can be used for fungi or other eukaryotic microbes like parasites and worms. So again, coming back to this tree of life. This was kind of 16S rRNA sequencing really served to help define it. So you can identify the 16S gene is present in bacteria and archaea. And in general, this 16S rRNA forms part of all bacterial ribosomes. So it contains regions of highly conserved and then highly variable sequence, so both of which are useful to the technique. So this is just showing the secondary structure and sequence of the 16S rRNA, and you can see these V regions shown in the diagram on the left are actually the variable regions. And this variable sequence can be thought of as kind of a molecular fingerprint. And so it's used to identify bacterial genre and species. There are these very large public databases that have been generated over time, available for comparison to your particular sample against these databases. And the benefit of the conserved regions are that you can actually use them to design primers so that you can successfully amplify across these variable regions. So one thing to always keep in mind when you're actually analyzing 16S data is that though you'll certainly end up with number of read counts from your analysis that fall into different bins of taxonomic identity. There can be copy number variation for different individual bacteria. So you can't make direct, you can of course guesstimate relative abundances, but that copy number variation can of course lead to variability in the actual abundance of any particular bacteria in your sample. This is just kind of a different schematic, also showing the 16S rRNA variable regions. So again, you can see the C regions and the V regions, just demonstrating the conserved and variable areas. So you can see there's kind of 9 variable regions that can commonly be used for 16S sequencing. In general, the V4 region has been kind of one of the most commonly used ones. That's one that my lab routinely makes use of. It's, fairly easy to make comparisons. There are kind of increasing approaches to try to capture all of the regions. And one challenge is that if you only sequence a single variable region, you don't get really great species level resolution. So you can get pretty confident resolution down to the genus. But then beyond that, it gets a little tougher. If you do kind of sequence additional of these regions, then you may have the capacity to actually gain some better species level resolution of your taxa. Okay, so in general, kind of the workflow for doing 16S, of course using next generation sequencing, would be to extract DNA from your sample, amplify your marker gene, in this case, perhaps the V4 region, using barcoded primers with different barcodes for each sample. You can then pool your amplicons and of course, sequence both the variable region and also your barcode. Then you can assign all of these different regions, again, such as the V4 region, to kind of a species, again, with the caveat that you might not get great species level resolution, but at least 2 taxa. And so that can include different definitions, which we'll get into, either operational taxonomic units or amplicon sequence variants. And then you might actually move on to performing visualization and comparison of different community relationships. And so he's just showing, one of the most classic examples of, if you look at samples from different parts of the human body, you see that the gut kind of clusters together versus the oral cavity versus a lot of other areas on the body which are more closely related. And so again, we'll kind of get a little bit more into what those sorts of graphs mean. Okay, so in just thinking about what a particular 16S read might actually mean, the definition of species is a little bit of a challenge. We've already talked about, the intense diversity, the potential for horizontal gene transfer, et cetera, when it comes to microbes. So that can be challenging. Obviously for eukaryotes, it's a much more kind of straightforward definition of, a group of organisms that can easily exchange, DNA. But in this case, microbes are a little trickier to pin down. So in general, if you think about, again, plasmids, horizontal gene transfer, transposons, viruses. There are all these complex things happening in bacteria that can make it really tricky to really define a species for amplicon data. But in general, again, with kind of all of those limits in mind, we can at least attempt to bin amplicon sequences and use them to at least estimate the relative abundance of different taxa in a sample using either OTU or ASV definitions. And so just because I think this is kind of a part of the field that's been evolving over the past few years. So, initially 16S data analysis was focused really upon defining operational taxonomic units, or OTUs. And so depending on kind of how you choose to cluster your sequences, then you can end up with different number of OTUs assigned for each sample. So for example, what you can see on this graph to the right here for OTUs is that if you're highly exclusive and say, okay, I'm only going to bin together V4 sequences that are 100% identical, then you'll end up with, just under 1,000 different OTUs in your sample. But, there's Illumina error, and there's, kind of been a shifting definition of how closely related are these different taxonomic units. And traditionally, a lot of people were actually serving to choose to bin sequences that were 97% similar or greater into the same OTU. And so in that case, then you might end up with something closer to, let's say, 600 OTUs. So of course, these definitions matter. And that's why I think in general, I can say the field has been gradually sort of moving away from these because, you might actually be losing a lot of the resolution as far as what taxa are really present in your sample. But in general, some of the most classic approaches for doing 16S analysis that were kind of long used led us to a lot of really critical, important findings in the microbiota, and of course, extremely valuable from that perspective. Include CHIME, Mother, and USearch. And so, if you're making use of any of those programs or bioinformatic pipelines, then in general, they'll tend to bin sequences in that way, depending on how you kind of set your parameters. So newer approaches are more focused around amplicon sequence variants. And so in this case, it's really attempting to reconstruct the exact biological sequences that are really present in the sample. So these are also called zero noise OTUs. And so kind of any sequence really is given its own ASV. So any downstream analysis really maintains that information about what exact sequences were present. And so of course you end up with many more ASVs than you would 97% similar OTUs when you're doing your analysis. So, depending on how analysis is performed, it can be a little bit more computationally expensive, but does seem to ultimately be likely more useful and more kind of biologically accurate. So approaches that are making use of that include Data2 and CHIME2. Okay, so and just thinking about how are we kind of defining species for this tag data? Of course, we search for sequence similarity between these variable regions. Of course, these genes are housekeeping genes, which are less likely to be involved in horizontal transfer. And so, these should be remain fairly well conserved. But as I mentioned earlier, these sequences, of course, can also have variable copy numbers, which can lead to some sort of biased results. So when we're actually trying to bin these tags, there are kind of two broad differential ways this can be done. So one is sort of not making use of a reference database, but instead using composition-based binning. So this is going to be most helpful if you're analyzing a sample in which you're actually expecting to find novel species. And this actually it may, of course, kind of make use of reference databases, but is more relying on comparisons of gross features to these as opposed to exact sequence equivalence. And so some of these features can include things like GC content, camera-based frequency comparisons, or codon usage statistics. If you're working with a sample in which you would expect that the majority of the different tags on there have probably are already present in a robust database, then you're more likely to use similarity-based binning. And in general, this would include direct comparison of your OTU or ASV to a reference database. The identity cutoff may vary depending on what kind of resolution you require. So again, there's kind of that 97% cutoff that people think of, used to kind of say, okay, these are likely similar species. But that's kind of a squishy cutoff. For things like genus and family, where you're not relying on quite as close of match for your particular tag, then it's much more comfortable definitive identification of those. So one challenge can be that if you're kind of being too stringent in your cutoff. Certainly, this is the case with OTUs. You can, and some of the programs that or bioinformatic pipelines that make use of these operational taxonomic units, you can run into the challenge that excessive diversity can end up being reported. And so, that's something like a sequencing error or a sample prep issue could make you falsely interpret that you have way more new interesting microbes present in your sample than you actually do. Okay, so in general, well, so just to make kind of one additional point here. So the use of barcoded primers for sexiness analysis is tremendously useful because it turns out that for well-characterized sample types, for example, like poop, where many people have sequenced a lot of poop a lot of times, and there are very robust databases for it. You can actually get away with doing relatively few reads per sample to get a pretty robust survey of your sample. So for some of our mouse poop analyses, we're able to get away with on the order of 1 to 10,000 reads per sample. And so if you think about doing a single Illumina run, you can obviously multiplex a pretty huge number of samples onto a single run and still get really solid coverage. Okay, so again, I kind of mentioned that some of the use of OTUs maybe starting to fall out of favor a little bit. And this is really kind of the most traditional CHIME workflow that, a lot of really important early microbiota data analysis was performed using. But this is maybe now starting to get to become a little less used and is evolving more towards the ASVs. But I'll still go ahead and go through the CHIME workflow just a little bit. And I will say that, it's still very useful. This was certainly the first pipeline that I ever made use of. And that was, whatever, 10 years ago. This was very state of the art. But, yeah, I'll leave it at that. So there's still a lot of people using CHIME, and it still is certainly a great way to get a broad sense of what texts are present in any given sample. So for actually analyzing 16S data, kind of with a lot of different pipelines, the key steps would be some early processing steps of your FOSQ file, removing primers, demultiplexing, quality filtering. Then there's the actual picking of the operational taxonomic units. So that could be based on a reference-based approach. So for CHIME, Green Genes is generally the database that it uses. And you then basically build an OTU table, which is just giving per sample what were the counts of that particular operational taxonomic unit that were present. And so just to kind of clarify, what would actually show up in that table would be Escherichia was detected in sample 1 at 10 counts, and sample 2 at 0 counts, and et cetera, et cetera. These pipelines can also do de novo OTU picking, in which case it's not actually initially making use of putting each sample against reference database, but actually serves to kind of characterize and look for novel, tries to bin the different variants of sequence prior to then trying to assign taxonomy. And so basically, these are just slightly different approaches that the pipeline would use. One kind of throws away things that can't be classified. The other would keep those things that can't be classified, and then would maybe give a very loose taxonomic assignment, or say it can't be assigned. And then you might realize that, oh, okay, there was actually some very novel variant that was present, or novel taxa that was present in any particular sample. So then once you have actually successfully obtained an OTU table, which is that sample per sample count table, and you also can get a phylogenetic tree that shows the evolutionary relationship between these different taxonomic units, not just based on the Green Genes database. Then you can actually go through and take advantage of these to look at things like alpha diversity and beta diversity, as well as visualization, which we'll get into a little bit more. Okay, so the kind of more recent approach, as I mentioned, looking more at ASVs. So some of the advantages of these amplicon sequence variants in lieu of OTUs are that they can help resolve biological differences of even 1 to 2 nucleotides. You'll end up with fewer false positive sequence variants than with other methods. The ASVs have the benefit of being directly comparable between studies without having to actually get the raw data and reprocess it, which is hugely beneficial because it kind of in the past used to be that, you know, depending on what parameters somebody had used, you know, a particular OTU count table might not actually be able to be directly used to compare to your data. And then additionally, the compute time actually scales pretty linearly with the sample number. So the .2 format, I hope it does not sound that I'm advertising for it, but our group has moved towards using this instead of the kind of original Chime, and Chime 2 actually incorporates a very similar approaches to that too. So in general, this is, I think, kind of where the field is moving for 16S analysis. So, you know, honestly, the pipeline is quite similar to the Chime one, wherein there's, of course, you know, quality control and filtering steps, dereplication to actually ensure that you get unique sequences, denoising, and then assignment of taxonomy and phylogeny. And then you end up with kind of an object that's very similar to the OTU count table that Chime would generate. And then you can move on to doing more downstream steps, including, again, alpha and beta diversity analysis. Okay, so kind of getting more into those. So whether you have a count table from OTUs or from ASVs, you know, fundamentally what you care about is how do some of my samples compare to some of my other samples. And so, you know, you can think of this in healthy versus disease samples. In our case, a lot of our things we're looking at are, you know, mouse that received this treatment versus mouse that received another treatment or no treatment. And so in general, kind of the two big things that are done with 16S data are looking at alpha diversity and beta diversity. So alpha diversity is really what is there, how much is there. So it's focused on what's the level of diversity present within a sample. And then beta diversity is actually looking at how do samples compare to each other and how similar are they. So focusing more on diversity between samples. Okay, so for measuring alpha diversity, this is, again, diversity within a sample. So there are a number of different indexes that are commonly used. Simpson's, Shannon, Chow. You know, ultimately, if there is, you know, one sample coming from a very non-complex state compared to one from a state where there's, you know, a much more abundant number of organisms, it really doesn't matter exactly which index you use, you'll end up finding that, you know, there's greater diversity in the more complex environment. If you're looking at some things that maybe have very subtle differences, then, you know, it might matter what specific index you use. So for example, the one shown here is just giving less weight to the rarest species. And so that's going to vary a little bit depending on what specific index is used. In general, people tend to probably look at them all because it's very straightforward to kind of do all these calculations simultaneously. So beta diversity is a little bit, at least for me, tends to be harder to kind of think about. But in general, it's just the diversity between samples. So this is just showing an example, which is Sorensen's index, where you're looking at the number of species in sample 1, the number of species in sample 2, and then the number of species that are actually present in both samples. And so there's just this, in general, the idea is that if there are a lot of the same species present in both samples, they're going to be more closely related. Whereas if there are, like very few species present in both samples, then they're going to be more dissimilar. And kind of one of the most commonly used metrics for actually looking at that at beta diversity is actually unifrac distance. So this is actually looking at shared branch lengths on phylogenetic trees. So it kind of takes that also into account. And in general, this distance metric for comparing between different biological communities actually serves to kind of then incorporate that phylogenetic information into how it provides these distances. So this is just showing kind of an example of 3 different communities, or sorry, of, well, yeah, 3 different scenarios. So with 2 different communities. So one community is red, one community is blue. So if the communities are exactly the same, so totally identical, and they have all of, every single species is present in both samples, then your unifrac distance is going to be 0. These are the same. In contrast, if every species present in red is absent in blue, in the blue sample, and they're just totally phylogenetically unrelated, then you're going to end up at a kind of maximum unifrac distance of 1. And that's just based on the fact that, or the idea that if a certain phylogeny has developed in a particular organism, then related, phylogenetically related other taxa are also going to emerge in that same habitat. And so you can just kind of take advantage of that to say, okay, well, if the habitat is conducive to growing certain phylogenetically related taxa, then another totally unrelated habitat is probably not conducive to those same taxa, and is in fact conducive to a different set of taxa. And so the more phylogenetically unrelated all of these taxa become, the more confident you become that they are really totally unrelated communities. And we'll get into a few examples to try to make that more clear. And if they're kind of in the middle, so, for example, something like a poop sample from one person versus a poop sample from someone else who lives maybe in another country, you can imagine that the human gut is a place where you probably see a lot of the same samples, but maybe dietary patterns or other environmental conditions might lead to there being some distinct taxa present. So you might say they're moderately related, but still, have some distinction. So that might be the kind of that more intermediate state for unifrac distance. Okay, and so this is just showing, now if we start to think about, okay, not just 2 communities, or sorry, not just 2 samples, but taking the complexity up a notch by looking at 3 different samples, if we're actually trying to cluster samples using this unifrac distance, then we can look at the interrelatedness of each sample to the other. So if we look at the red versus yellow, we can say, oh, okay, well, there actually seems to be a pretty decent amount of overlap. There's some phylogenetic relatedness between the species that are present, between the red and the yellow. So they're actually, fairly well related. If we look at red versus blue, they look almost all different, but okay, there's, at least one species and the red is clustering with the blue. So, all right, there's like a, smidge of relatedness. And then the yellow versus blue just look completely nothing to do with each other. So then basically you'll calculate this distance matrix comparing all of the samples against all of the other samples. And then what you could do is either use that to build a hierarchical cluster, where again we see red and yellow samples look more closely related to each other than either of them do to blue. Or you can look at this on the sort of 2D plot here, which is a principal coordinates analysis, where you can actually see how closely grouped these different samples are. So because red is more similar to yellow, it ends up closer together in this principal coordinates analysis plot. The red is slightly more similar to blue than blue is to yellow. So you can also see that they have less, the red and blue have less distance between them. And so this is just kind of a fairly intuitive way of looking at the interrelatedness of samples to each other. And, while this is perhaps not super useful for looking at how 3 samples compare to each other, this is very useful when you're looking at a large number of samples. Okay, and so just kind of showing an early example of 16S community analysis. This was actually done using 454 sequencing, so a little bit of time ago, but actually from 2008, from Jeff Gordon's lab, who, of course, I'm sure everyone is already aware, really pioneer in the microbiota field. This was a really interesting study looking at the effects of diet on the microbiota. And so focusing on 3 main groups, which are carnivores, herbivores, and omnivores, with the idea that, the different nutrient composition of diet may, of course, lead to very differential communities present in the gut. And so here you can see kind of the most fundamental finding here, which is that mammalian microbiomes cluster by diet, which I think, now is not that shocking, but at that time was something that was, kind of a surprise that this would be so clearly true. So in just looking at this kind of very complex principal coordinates analysis, what you can see is that the circles represent different mammals. Of the different types, so either again herbivore, carnivore, or omnivore, based on the coloring, and then depending on the thickness of the line, there's either more or less shared sequences. If there's a line, then between the circles and then these gray rounded squares, that indicates that there was an OTU found. And then the diamonds, which are in the periphery, are actually OTUs that were unique to individual samples. And the number of unique OTUs in this case, it gets bigger with the more unique OTUs that are present. And what you see is that the carnivores cluster together really nicely. The 2 different types of herbivores, the foregut and the hindgut fermenters, cluster together somewhat, but also separately. And then omnivores are kind of all clustered together, including humans, despite, of course, diverse habitats and diets. And so just suggesting that, actually the way that we kind of process our food and choices of what the means are by which we actually perform digestion, then will lead to extremely different microbial communities present in our guts. So, in general, of course, these sorts of approaches have been terrifically important for kind of a number of different observations or scientific discoveries. So one of the, this is kind of a key one, focused on looking at healthy individuals versus patients with inflammatory bowel diseases. So here you can see differential clustering of healthy individuals versus those with Crohn's disease or ulcerative colitis. And this is just kind of an example of how that can be visualized, but this is a very well-supported observation that's been found by many, many different groups in general, that people with inflammatory bowel disease have lower bacterial diversity, and they also tend to have outgrowths of different microbes that can serve as pathobionts, and in general tend to cluster quite differently in principal coordinates analysis. Stickiness analysis has also been used to demonstrate that you end up sharing a reasonable amount of microbiota with your dog. And this is just kind of, I think, more of a fun example, but this particular graph, they're taking advantage of the principal components or coordinate analysis to show, and sorry, this is just one that's also plotted in 3D to look at three components simultaneously. But what you can see is that so in general, the study showed that individuals with dogs tend to have taxa that are different from those without dogs, largely due to the presence of dog-derived bacterial taxa on their skin, presumably from pretty frequent contact. And interestingly, what you can see, so the central, or sorry, the plot on the left kind of shows all of the samples. But then the two plots on the right, if you kind of look back and forth between them, what you can see is that the dog gut and tongue communities actually harbor microbial communities that are somewhat similar in diversity and composition, whereas that's not the case for the human tongue, and human stool is much more distinct. So those are some kind of interesting observations from that study. And then another really, I would say, kind of paradigm shifting study that came out of Jeff Gordon's lab in 2012, was looking at the acquisition of the human microbiota over time, and also in the context of very different geographies. And so this particular study had a lot of very cool fundamental findings, again, that have held up to multiple different types of, or multiple similar kind of related studies have shown very similar findings. So, these are very fundamental observations that have also been supported by the passage of time, always reassuring. But in general, kind of the first graph is showing that in general, the phylogenetic composition of bacterial communities tends to kind of become more adult-like over that first 3-year period of life. And again, this is focusing on those unifrac distances. shown earlier. So while children in general will start off very different from each other, the adult configuration and also just even the older child configuration tends to be much more closely related. In addition, they found that this observation that variation among children compared to adults was robust to geographies. This is true kind of regardless of the geographical area that you're looking at. And then also they found that, again, something that we now have much greater appreciation for is that the phylogenetic composition of the fecal microbiota is very different between individuals living in different countries. As you can see, the US really clusters quite separately from Amerindians or Malawians. And so these kind of 16S surveys are certainly have been extremely important for performing a number of really fundamental studies looking at health and disease in humans. Okay, so the last thing I'll be talking about in this lecture is focusing on sequence-based metagenomics of and looking at shotgun environmental sequencing to characterize genetic diversity. So I'll give a little bit of background and then just dive into a few examples, and then we'll wrap up. I'm sure much to your relief. So we kind of already went through how 16S serves as the tagging, but there's also the option to just do shotgun sequencing to really get a sense of the community that's present. So the method that kind of commonly employed for that would be for any individual sample, extract the total DNA from your mixed microbial community, shear it and select for particular size fragments that are practical to sequence, and then barcode them. And then you can do that for as many samples as you like, put different barcodes on, pool these, sequence them, bin your reads based on the barcode, map these to whatever database you can, and then you can either use these to generate count tables that are related more to the who is there, or count tables related to what is there as far as particular genes. So just starting off with example one. So this is kind of a really interesting study that was done by Craig Venter, in which whole genome shotgun sequencing was performed on these microbial populations that were collected from the Sargasso Sea near Bermuda. And this shotgun sequencing, again, was happening quite a while ago, but certainly at that time, there was just a huge amount of microbial diversity that was totally new. So from this study, they were able to identify, because they did shotgun sequencing, 148 new 16S rRNA sequences, and those were specifically bacterial phylotypes, but those from what they actually found were 1,800 approximately different species that then we're kind of mapping onto these new as well as known bacterial databases. And so, using shotgun sequencing, especially of these kind of interesting and maybe less explored environments, obviously there's, since then been quite a bit more studies done in marine environments. but there continue, of course, to be many habitats that are totally unexplored. That was able to, really add a huge amount of information to databases. That was then followed up by a wider global ocean sampling expedition. So they really found a huge number of additional data, or a huge amount of additional data. So we're able to double the total number of known proteins, able to get coverage of really approximately all known prokaryotic protein families, and just kind of continuing to build these databases is really helpful because there had previously been kind of 6,000 genes that had already been in the literature that were orphans. Basically, they had kind of been shown up but had no known homologs. But then it turned out that they did have homologs in this global ocean sampling data. So in general, this shotgun sequencing approach in these particular environments was, of course, very useful. Okay, so in addition to kind of expanding, just the amount of whole genomic coverage and information that we have about genes that are present in different environments, shotgun sequencing also can potentially be highly beneficial to clarify community structures and metabolism through even kind of simple environments. So in this case, this is from a study doing shotgun sequencing of DNA from this natural acidophilic biofilm. And they were able to actually, because it was a pretty simplified microbiota, they're actually able to assemble genomes for all 5 lineages and end up with kind of this really rich data that gave huge insights into survival strategies that might be used in these this particular extreme environment. And so this is an example where shotgun sequencing, in addition to capturing wide varieties of different microbes, can really certainly be highly beneficial in a smaller community because you can really get nice resolution of the contributions of each individual bug. So to kind of take that a step further, there's also progressively become a lot of been gaining interest in kind of the microbiota / metagenomic field has been in single cell sequencing of these uncultured organisms. Because then you can really end up with fantastic genomic resolution without having to figure out how to actually cultivate some of these bugs that, or many of these bugs that might be extraordinarily challenging to grow. So an example, and this is, kind of started as far back as 2006, was trying to either use a substantial dilution of a complex community or single cell sorting, then lysing what you hope is kind of an individual bacterial cell, then subjecting the DNA that's present to multiple displacement amplification. And so you can see kind of 2 different schematics of this, both in the diagram on the left and then also the slightly redundant one on the right. We're using this 529 enzyme to kind of and as well as random primers to amplify your genome as individual strands continue to grow by polymerization, then those strands get displaced, new primers bind, so that newly formed DNA, and there's kind of a progressive build of this complex hair tangle of DNA. But the benefit of this multiple displacement amplification is that it actually can amplify the cellular DNA, again, from even one single bacterial cell, greater than 10 to the ninth fold. And so then you actually end up with a practical amount of material that can be used for shotgun sequencing. And so once you end up with this kind of complex tangle, then you can end up digesting it, and then using different shearing and enzymatic treatments, and then you can go ahead and barcode and sequence it. And so in this particular example, just showing the distribution of sequencing depth of two different individual cells that were studied using one of these methods. And in general, what you can see is that even though these were done with multiple displacement amplification, they ended up getting nice sequencing data from greater than 60% of the genome. And it, that was kind of across the genome. And then once you kind of have that level of resolution, then primers can also be designed from the sequenced areas in order to sequence across some of the less well-resolved areas of the genome. And so this technique of single cell isolation and then multiple displacement amplification and sequencing has been used to sequence a number of different archaeal and bacterial cells from diverse habitats. And I think this is kind of tip of the iceberg. There is obviously a huge amount of utility in being able to do this. And with each of these genomes that gets nicely resolved, then that in turn leads to better resolution of other metagenomic samples, because there can be more, each individual read from shotgun sequencing can be more easily assigned back to an original genome. And I'll just mention this recent paper because this is within my area of interest, looking at phage bacteria interactions. And so this has even recently been applied to start to get at this, where from this paper, they actually divided human stool samples into viral and bacterial fractions, stained the viruses, and then combined them with the unstained bacteria, sorted them, performed MDA, and shotgun sequenced. And then they were able to get the bacterial genomes, of course, which has been kind of the main thing we focused on so far. But they can also get the phage genomes, and they get information about which bacteria the phage was actually infecting. So kind of get all three in one nice single genetic analysis. So, I think there is also going to continue to be a lot of creative approaches that are going to be taken to help resolve a lot of the dark matter that still remains in the microbiota. Okay, so just as kind of the last example, increasingly as sequencing gets cheaper, we're getting more information and more complete genomes from metagenomes. And these apply to these really kind of interesting and unusual sites. So there have been some cool metagenomic analyses of permafrost, where they've actually been able to get really nice draft genomes from these, from complex metagenomes. And permafrost is kind of a super interesting particular habitat, because you can look at over time, as things have shifted from frozen to thawed states, how has that changed communities? Additionally, there are obviously a lot of interesting things to be found, kind of in any area that you look at. So this is just an example from looking at cow rumens. And just, from these sorts of basic analyses, they've been able to assemble 15 uncultured microbial genomes. And I'll just mention that increasingly there have been better bioinformatic tools that are in development to bin microbes into individual genomes from metagenomic data, also called metagenome assembled genomes. It's still often a challenge to circularize and actually complete these genomes. So draft or partial genomes are commonly submitted to databases. But even these partial genomes are still extremely useful for comparing new sample sets, and actually trying to identify different functional gene sets that are in common. Okay, so also speaking of functional gene sets, in the next lecture we'll be talking more about function-based metagenomics. And with that, I will wrap up. I'll say if you have any questions about the content covered today, please just email me or ask your very excellent TAs. And I, of course, I'm happy to try to clarify anything or provide more information or reading materials if you're interested. I hope that everyone stays well, and I will be planning to post the next lecture in a couple of days. Great. All right. Thank you.